Welcome back to the Translational Addiction Podcast. In our previous episode, we talked about how nicotine administration in adolescence and the fetal period can be detrimental to brain development. We explained how teenagers are uniquely vulnerable because their brains are still in rapid growth, synaptic pruning, and remodeling of reward pathways. Nicotine experimentation during teenage years disrupts developmental processes, overstimulating dopamine circuits, rewiring habit forming networks, and weakening attention and emotional regulation in ways that are more severe and irreversible than in adults. In today's podcast, we will investigate addiction liability across individuals and why some people may be more prone to nicotine addiction than others throughout their lives. We will focus on a clue that might be the missing genetic link in understanding this as we explore the review article. Unraveling the role of CHRNA6, the neuronal alpha 6 nicotinic acetylcholine receptor subunit, by Yasamin Haydari, Emily Castro, Shahra Dolfipour, and Frances Leslie.
In 2021, an estimated 46 million adults in the US reported daily use of tobacco or e-cigarettes. But what's more concerning is the youth data. Over 6 million middle and high school students reported experimenting with tobacco products in 2022. In our podcast covering the review Nicotine in the Developing Brain, we explained the concept of the gateway effect, which is how nicotine use can increase the likelihood of experimenting with more serious drugs of abuse later in life, which is why teen nicotine exposure is such a concerning public health issue. It is crucial to understand the molecular mechanisms explaining nicotine action and how nicotine dependence develops. This brings us to our focus today, the alpha 6 nicotinic acetylcholine receptor subunit.
This subunit is unique because its expression in the brain actually peaks during adolescence. Preclinical studies have linked alpha 6 containing nicotinic acetylcholine receptors to nicotine-induced locomotion, dopamine release, and self-administration behavior. There's a significant genetic component, which researchers have identified as a specific single nucleotide polymorphism, or SNP, in the 3' untranslated region of CHRNA6, the gene that codes for this subunit. This polymorphism is consistently and strongly linked to increased smoking, higher rates of daily cigarette use, and an overall increased susceptibility to substance use in humans, especially during the critical development window of adolescence. Welcome to the Translational Addiction Podcast, where we decode the innovative research from the Lotwipur Lab in UC Irvine, dedicated to translational addiction studies and developing effective treatments. I am Thanvi Garnani. I am one of the hosts of this podcast, and I'm here with Michaela and Ardavan.
I'm Michaela Samortin, and I'm also one of the hosts. I'm a fourth year at UC Irvine.
I'm Ardavan Mehrozma, and I'm a third-year biological sciences undergrad at UCI, and I'm happy to be here with our two other brilliant co-hosts.
Thank you. So we have referred to nicotinic acetylcholine receptors, or NAChRs, as a vital functional unit regarding nicotine addiction in previous podcasts, but this review explains the structure of these receptors, which will assist in our understanding of their function and why so little research has been conducted on the alpha 6 subunit specifically up to this point. So functionally, NAChRs belong to a family called pentameric ligand-gated ion channels. Think of the receptor as a hollow 5-sided barrel embedded in the membrane of a neuron. That barrel is built from 5 individual protein subunits that wrap to form a hollow cylindrical structure. The lid of this barrel, or the gate, only opens when a specific chemical key called a ligand binds to it. So that ligand can be the brain's natural neurotransmitter acetylcholine, or it can be an external compound such as nicotine. Each subunit spans the length of the cell membrane. So when a ligand binds, the gate of these subunits open, allowing ions such as calcium, potassium, and sodium to flow through the channel and into the neuron. This sudden ionic flux alters the neuron's electrical state, either triggering or modulating neuronal signaling. Nicotine can also bind to NAChRs, which can disrupt endogenous acetylcholine activity and may lead to increased responses from a cell. The 5 subunits that form the channel are very diverse. There are 9 alpha subunits, alpha 2 through alpha 10, and 3 beta subunits, beta 2 through beta 4. So, different combinations of these 5 subunits form distinct receptor subtypes, and each specific assembly acts like a functional recipe that determines how strongly nicotine binds, how quickly the receptor desensitizes or shuts off, and what downstream signaling pathways are activated. Broadly, nicotinic acetylcholine receptors fall into 2 assembly types. So the first is homomeric receptors, where all 5 of the subunits are identical, and this is, for example, the well-known alpha 7 receptor is a common one of these. And secondly, there are heteromeric receptors, which have various combinations of the alpha and beta subtypes, and these are much more common and diverse. In most heteromeric receptors, including the alpha 6 containing receptors we're focused on, the ligand binding sites are formed at the interface between an adjacent alpha and beta subunit. This highly specific architecture gives each receptor subtype its unique pharmacological fingerprint.
Thank you, Michaela, for introducing alpha 6 subunit. Now I think it's a good time to ask, where is exactly alpha 6? What really sets alpha 6 containing nicotinic acetylcholine receptors, or alpha 6 NAChRs, apart is their extremely restricted expression compared to far more abundant NAChR subtypes in the brain. The messenger RNA for the alpha 6 subunit, or CHRNA6 gene, is primarily expressed in just a few highly specific regions deep within the midbrain, which are the following: the locus coeruleus, LC, the substantia nigra, or SN, and most importantly for our discussion, the ventral tegmental area, or VTA. The VTA is the core circuitry underlying reward, motivation, and reinforcement in the mesolimbic system, which are primary circuits that get hijacked by addictive substances. Nicotine's addictive impact depends on activating dopaminergic neurons in the VTA, which then project to downstream targets such as the nucleus accumbens, or NAC. Foundational studies show that alpha 6 transcripts are co-expressed with alpha 4, beta 2, and beta 3 subunits in roughly 90% of VTA dopamine neurons. This is like very exactly where they need to be to cause trouble. Alpha 6 subunit expression was previously believed to be restricted to VTA dopaminergic neurons. However, recent data show that alpha 6 transcripts are present in roughly half of both GABAergic and glutamate neurons within the VTA. There is a major conceptual shift in how we perceive alpha 6 receptor location, function, and significance. It means that alpha 6 receptors aren't just modulating dopamine output directly, they're also regulating the inhibitory, which is like GABA neurotransmitter, and excitatory, which is glutamate, inputs that shape the entire reward circuit. Functional alpha 6 expression has now been detected in non-neuronal cells as well, specifically astrocytes within the VTA. In mouse model, alpha 6 is co-expressed in astrocytes, which are the star-shaped glial cells, alongside alpha 4, beta 2, and beta 3 subunits. For decades, astrocytes were viewed as passive support structures, but we now have molecular evidence suggesting they may directly participate in cholinergic signaling relevant to substance use disorders, which involve drug seeking, extinction, and relapse. Studying age and sex differences in alpha 6 expression is critical to evaluate, since adolescence is a time of vulnerability to nicotine use. Studies show higher relative alpha 6 expression in VTA astrocytes of adult males compared to adolescent males, indicating that the cellular environment itself changes as the brain matures. Given that adolescence is a peak window of vulnerability, these shifts in expression are critically important. 6 differences have been observed in the neuronal excitability of the MHB, or medial habenula, with adult males exhibiting heightened excitability compared to females when exposed to nicotine and menthol. High-dose nicotine combined with menthol, highly relevant in modern vaping, may reduce the number of functional alpha-6, beta-2, beta-3 receptors within this nucleus. Such a reduction could increase neuronal excitability within aversion circuits, offering a possible molecular explanation for 6 specific differences in response to flavored nicotine products. Taken together, alpha-6 receptors are not limited to catecholamine neurons, but are also expressed in a wider variety of neuronal and non-neuronal subtypes, like astrocytes, opening new avenues for therapeutic targeting. It suggests cessation therapies may vary in effectiveness based on sex, age, and product type. These insights make it clear that future addiction research must move far beyond from a simple dopamine neuron model and instead account for entire circuits, including glial cells, developmental stage, and biological sex.
Thank you, Ardavan. So why is studying the alpha-6 subunit so challenging? For decades, the biggest obstacle wasn't a lack of interest. It was a profound lack of effective tools. Tools had not been created to study alpha-6 containing NAChRs due to its extreme low levels of expression and lack of selective agonists or antagonists. Essentially, it was not only difficult to find alpha-6 NAChRs in the body because of their scarcity, but because there was no good tool to target them. Additionally, the alpha-6 subunit shares roughly 61% amino acid sequence homology with the highly prevalent alpha-3 subunit. This level of genetic similarity made chemical distinction between alpha-6 beta-2 and alpha-3 beta-2 receptors nearly impossible. So where did the breakthrough in investigating alpha-6 come from? The cone snail venom. These tiny predatory marine mollusks can produce an extraordinarily potent venom which contains a cocktail of peptides known as alpha-conotoxins. Researchers have isolated and characterized these toxins to find ones with extreme selectivity. For example, alpha-conotoxic MII and its engineered analogs show high selectivity for alpha-6 containing receptors over the closely related alpha-3 beta-2 subtype. Even more critically, alpha-conotoxin PIA was the first toxin to show reliably be able to discriminate alpha-6 containing receptors from non-alpha-6 receptors. While alpha-conotoxins are phenomenal experimental tools, their in vivo utility is really limited. They are relatively large peptides, which are often unstable in circulation, and more importantly, they do not readily cross the blood-brain barrier. Now, another major bottleneck in drug discovery was the inability to express functional alpha-6 containing receptors in vitro because of a fundamental flaw in the receptor's assembly and trafficking process. When researchers initially tried to express alpha-6 containing receptors in standard lab cell lines, the subunits simply misassembled or aggregated. Receptor binding was weak and surface expression was minimal. However, new findings have shown that each alpha-6 NAChR's composition requires specific protein machinery to aid its assembly and transfer onto the membrane. If you are interested in learning more about this machinery, it's explained in way more detail in the complete review article that we're going over. While drug discovery advances, animal models have been essential for establishing function. Genetics have been absolutely pivotal here, so the simple and most informative model is the alpha-6 knockout mouse, which is genetically engineered to completely lack the alpha-6 subunit. As for visualization, by tagging alpha-6 with GFP or green fluorescent protein, researchers have been able to achieve high-resolution anatomical mapping. This model confirmed unexpected findings like alpha-6 expression in astrocytes. But the most compelling evidence comes from gain-of-function models. The alpha-6 hypersensitive knock-in mouse is particularly striking here. Researchers introduced a single point mutation to the alpha-6 subunit that makes the receptor far more sensitive to nicotine, so it activates more easily and it remains open longer. These mice show dramatically enhanced nicotine-induced locomotor activity, even at extremely low subthreshold doses. This finding shows that the hypersensitivity of this receptor subtype is enough to profoundly alter brain output. It definitely cements alpha-6's role as a master switch for nicotine-driven behavior. The most sophisticated model has been the humanized rat. A specific human CHRNA6 SNP has been linked to addiction risk, But how do you study that mechanistically? Researchers created transgenic rats carrying the human version of the CHRNA6 3' untranslated region, or the 3' UTR, and they introduced either a GG or a CC variant. Importantly, the coding region of the gene was left unchanged. Although the 3' UTR is non-coding, it controls translation efficiency, directly influencing how much alpha-6 protein is produced. The clinical and preclinical findings for adding these genetic variants will be discussed later in the podcast. Computational modeling and multi-omics approaches are also indispensable for such a complex. In silico screening allows researchers to use known selective ligands as templates and virtually test thousands of compounds against alpha-6 containing receptor structures, dramatically reducing time and cost. This approach has already identified multiple promising alpha-6 selective antagonists. And finally, these omics approaches solve the low expression problem I was mentioning. When protein levels are too low to measure reliably, you measure the genes that can code for them. A couple examples are transcriptomics, which looks at RNA sequence, and ribosome profiling. Ribosome profiling is especially powerful because it lets us know not just which RNAs are present, but which are actively being translated in that moment. These tools have been crucial for uncovering age and sex-dependent differences, not only in alpha-6 expression, but across dopamine, inflammatory, and stress-related signaling pathways.
Thank you, Tempe. So now that we have gone over what alpha-6 containing nicotinic acetylcholine receptors are, where they are, and how we study them, we can answer a bigger question. What do they actually do in the body? So how do these alpha-6 NAChRs drive the core properties of addiction? A large body of evidence shows that these receptors are critical modulators of nicotine-induced dopamine release, and they do so with regional specificity. Alpha-6 NAChR effects are concentrated in the ventral striatum, particularly in the nucleus accumbens, which is one of those reward regions altered by addiction that we've discussed in previous episodes. They play a much smaller role in the dorsal striatum, which is more involved in motor planning and habitual behavior. That selective engagement of the reward hub highlights their central role in early addiction. The posterior VTA shows significantly higher baseline expression of alpha-4, alpha-6, and beta-3 subunits compared to other parts of the VTA. So how did they prove this region is essential? Researchers microinjected an alpha-6 selective antagonist alpha-conotoxin, PIA, into the posterior VTA, or ventral tegmental area. So when they did this, nicotine-induced dopamine release in the nucleus accumbens was completely reduced. This experiment provided substantial proof that alpha-6 containing receptors in the PVTA, or posterior VTA, are the gatekeepers of nicotine's acute rewarding effects. Blocking the receptor freezes the reward signal. Let's explore that neurochemistry on an observable level. A common behavioral proxy for stimulant effects in animals is locomotor activation, increased movement. And alpha-6 receptors are both necessary and sufficient for nicotine-induced locomotor activity. So when researchers partially inhibit alpha-6 function, nicotine-induced locomotion is reduced. Therefore, alpha-6 is necessary for nicotine-induced locomotion. Alpha-6 hypersensitive knock-in mouse, where a single point mutation makes the receptor more responsive, shows dramatically enhanced locomotor activity in response to nicotine. One hyperactive receptor subtype is enough to drive the behavior. When researchers infused an alpha-6 selective antagonist directly into the posterior VTA, nicotine-induced locomotion vanished, along with dopamine release. Block the receptor in that one location, and the behavioral output disappears. Therefore, alpha-6 is sufficient for nicotine-induced locomotion. Voluntary drug taking, choosing to seek and consume nicotine, is the most compelling evidence of addiction relevance. In self-administration paradigms, wild-type mice readily learn to press a lever to receive nicotine. In contrast, alpha-6 knockout mice completely fail to self-administer nicotine even when the dose is increased. Researchers took those alpha 6 knockout mice, animals that showed no interest in nicotine, and selectively reintroduced them, the alpha 6 subunit, only into the VTA dopamine neurons, and nicotine self-administration was fully restored. This proves that alpha 6 is a non-negotiable regulator of the nicotine reinforcement.
Nicotine dependence is highly heritable, with estimates ranging from 40 to 70%, so it's no surprise that genes encoding these receptors are key susceptibility candidates. The spotlight is on CHRNA6, particularly a single nucleotide polymorphism that sits in the UTR region we were discussing. This SNP is associated with nicotine dependence, greater number of cigarettes consumed per day, and a more positive subjective response to nicotine, especially when its use begins during adolescence. While the G allele is typically identified as the risk allele, some population-specific studies suggest it might be protective in certain groups, highlighting the complexity of some of these genetic findings. So, rats engineered with either the GG or CC variant of the human untranslated region allow for direct testing of how a single genetic difference, even if it's in a non-coding sequence, can fascinatingly alter behavior. Nicotine exposure produces increased locomotor activity that Michaela was talking about, and reduced anxiety in female CC rats and male GG rats relative to controls. So the same drug produces different outcomes depending on sex and genotype. Adolescent male GG rats show a significantly higher tendency to relapse into nicotine-seeking behavior after abstinence. Evidence suggests that the CHRNA6 GG variant increases susceptibility due to cue-induced relapse, a major predictor of cessation failure. A fascinating outcome also appears before drug exposure. Adolescent male GG rats had significantly elevated baseline dopamine levels in the nucleus accumbens score compared to adult GG males and adolescent CC males. So the reward system is already more primed to be susceptible. One of the most striking truths of addiction science is how tightly alcohol and tobacco use are linked. People with alcohol use disorder are far more likely to be nicotine dependent and vice versa. But why? When researchers injected mecamolamine, a non-selective nicotinic acetylcholine receptor antagonist, directly into the ventral tegmental area, it blocked alcohol-induced dopamine release in the nucleus accumbens. That result showed that alcohol's rewarding effects depend on nicotinic receptors, even though alcohol itself isn't a direct activator of those receptors. Human genetics studies then reinforced this link, showing that variation in CHRNA6, the gene encoding the alpha 6 subunit, is associated with both smoking and alcohol-related behaviors. What makes the alpha 6 subunit especially compelling is how it explains alcohol's dose-dependent effects on our reward circuitry. At low doses, alcohol increases dopamine release, but only when alpha 6 containing receptors are functional. Blocking these receptors collapses that dopamine signal. Animal models with hypersensitive alpha 6 receptors show greater alcohol intake and stronger reward learning, which reinforces alpha 6's role as a shared molecular level for both nicotine and alcohol.
The alpha 6 subunit is clearly a gatekeeper for addiction and reward, but it also plays a role in neurological disorders, including Parkinson's disease. But how does a receptor so central to addiction become relevant to neurodegeneration? Parkinson's disease is defined by the progressive loss of dopaminergic neurons in the nigrostriatal pathways, which projects from the substantia nigra pars compacta, or SNC, to the striatum. Importantly, the substantia nigra is one of the three brain receptor regions where the alpha 6 expression is highest. So the neurons that degenerate are rich in alpha 6 containing receptors, meaning that patients with Parkinson's disease are losing neurons with alpha 6. These dopaminergic neurons express both alpha 4 beta 2 and critically alpha 6 beta 2 containing nicotinic acetylcholine receptors, or AChRs. The degeneration of this pathway and the loss of these receptors underlies the hallmark motor symptoms of Parkinson's disease: bradykinesia, rigidity, and tremor. The standard treatment for Parkinson's disease is L-DOPA. L-DOPA is a dopamine precursor that the brain converts into dopamine, and it's highly effective, especially early on. However, long-term treatment is frequently complicated by a side effect known as L-DOPA-induced dyskinesia, or LID, characterized by involuntary, uncontrolled movements. Nicotine exposure has been shown to reduce L-DOPA-induced dyskinesia, but this predictive effect is completely absent in alpha 6 knockout mice. Even more compelling, studies using the alpha 6 hypersensitive knock-in mouse suggest that alpha 6 beta 2 containing receptors are directly involved in the development of dyskinesia itself. In other words, alpha 6 receptors play a functional role in both the disease circuitry and the side effects of its treatment. So alpha 6 becomes a key target for optimizing Parkinson's therapy. And this brings us to one of the most promising therapeutic strategies in the field: positive allosteric modulators, or PAMs or PAMs. A PAM or PAM does not activate the receptor directly. On its own, it does nothing. Instead, it enhances the receptor's response only when the brain's natural neurotransmitter, acetylcholine, is present. This dramatically reduces the risk of overstimulation and side effects, the kind you expect from a direct agonist like nicotine. Importantly, PAMs often show much greater receptor subtype selectivity compared to, for instance, nicotine. Is there a PAM specific to alpha 6? Yes. A compound known AN-6001 has been identified as a selective alpha 6 beta 2 nicotinic receptor PAM. In preclinical studies, AN-601 has been shown to protect dopaminergic neurons from neurotoxic damage, suggesting strong neuroprotective potential in Parkinson's disease. AN-601 shows a significantly higher preference for alpha 6 beta 2 receptors over alpha 4 beta 2 receptors, which are among the most abundant nicotinic receptors in the brain. So it selectively only targets alpha 6 compared to more abundant receptors. Now let's broaden the scope again and look at the chronic pain, another area where existing treatments are often inadequate. Chronic pain is frequently associated with mechanical allodynia, a condition where normally non-painful stimuli like light touch are perceived intensely painful. Studies have shown that a specific receptor subtype, alpha 6 beta 4 containing NAChRs, is expressed in the dorsal root ganglia, or DRGs. These neurons are the primary sensory afferents that transmit information from the body to the spinal cord. Experimental evidence shows that nicotine's ability to alleviate mechanical allodynia is mediated largely through these alpha 6 containing receptors. Alpha 6 containing receptors appear to directly interact with and inhibit P2X3 receptors, a class of ATP-gated ion channels that play a major role in neuropathic pain signaling. Mirroring the addiction findings, a specific SMP located in the CHRNA6 promoter region has been associated with altered pain sensitivity. Individuals homozygous for the minor allele were significantly more likely to experience persistent pain 6 months after surgery. The data strongly suggests that alpha 6 beta 4 containing receptors are functionally involved in pain modulation. This opens an exciting therapeutic avenue, one that moves beyond opioids and instead targets the receptor level mechanisms governing pathological sensory amplification.
Thank you, everyone, for joining this episode of the Translational Addiction Podcast. In conclusion, our investigation of the alpha 6 nicotinic acetyl receptor underscores a remarkable development in the field of translational addiction, which was shockingly facilitated by the precision of marine cone snail alpha conotoxins. We have moved beyond the traditional view of alpha 6 as solely a dopaminergic marker, as we now recognize its critical presence across GABAergic, glutamatergic, and noradrenergic neurons, as well as in astrocytes. The therapeutic implications are vast. Targeting alpha 6 offers robust pathways for mitigating nicotine and alcohol reinforcement, providing neuroprotection in Parkinson's disease through positive allosteric modulators like the AN6001, and addressing chronic pain via non-opioid signaling. Furthermore, identifying CHRNA6 gene variants allows for personalized addiction treatments, potentially optimizing clinical responses to therapies like varenicline. As alpha-6 expression peaks during the vulnerable period of adolescence, the challenge for public health is to develop precision prevention strategies that account for the complex interplay of genetic, sex, and cellular diversity. Understanding the alpha-6 receptor is like finding the master key to a complex mansion. It doesn't just open one door to treatment, it unlocks an entire wing of possibilities for protecting those most at risk. If you are interested in learning more about the alpha-6 subunit functions in addiction, follow us on Instagram at Translational Addiction Podcast for updates, and check out the infographics and papers on the Lotfipor Lab website linked in the show notes. If you or someone you know is struggling with addiction, you are not alone. Please contact your healthcare provider or refer to the resources linked in the show notes. The Translational Addiction Podcast is a production of the Lotfipor Lab at UC Irvine, exploring how to understand addiction and develop effective treatments. See you next time, and thank you for listening.